This is a production of Cornell University.
Let's see if this microphone works right. Okay. Thank you, Chelsea. I always tell everyone that the moment I first heard of Cycads changed my life. And this is the first time I get to say this in the room with Chelsea. I first heard of Cycads in her plant morphology course at UC Berkeley. And she said at the end of that, she was talking about all the different amazing things that I'm going to tell you about Cycads. And she said, if you're more interested, you should go read Island of the Colorblind by Oliver Sacks. And I literally stopped on the way home at the UC Berkeley library, which did not have it, I have to say. So we'll have to check Cornell. And then stopped at the Berkeley City Library and got that book that day. And it definitely was a change in my life. I've fallen in love with Cycads ever since. My background is that I've always been interested in plant-insect interactions, particularly from an agricultural perspective. My background is working a lot in sustainable agriculture before Chelsea changed my life. So it's okay. It's great, I think. I hope everybody likes that Chelsea changed my life. So I always like to start with this picture for a number of reasons. I think that right off the bat, I'm used to speaking to really diverse audiences, and so their first question is, what is a cycad? So I like this picture because it tells you a couple important things about the biology of the system. Cycads are gymnosperms, which I don't have to tell this room, means that they have cones as their reproductive structures, and they're also dioecious, so the female cones or the ovulate cones are housed on separate plants than the pollen cones or the male cones. And here I am looking at a female plant, I'm looking at all of these cones. But also I love this picture because it tells a story that matters to me. This was at a field season in Florida that I went with my husband. I have, you can see my wedding ring that was subsequently stolen when I was mugged in Brazil at the Evolution Conference. And I think those two things, thinking about relationships and thinking about the history and the importance of relationships and thinking about these plants really come together to like give a good introduction to what I'm gonna talk about today and really what I talk about always. I think that relationships are inherently interesting to all of us, and I always say that's evidenced by the success of every ridiculous romantic comedy movie ever made, no matter how silly the storyline or insane the outcome is. And that makes a lot of sense to me because we're curious, we are innately curious, like, to answer questions about these relationships. And these are some examples of movies that I love. If none of you have ever seen this or read the book, this is Lonesome Dove. It's an amazing, epic miniseries. If you know anything about me deeply, this movie sort of and book influences my whole life. I have like a handmade purse of Lonesome Dove. I love this movie. So please all go watch it when you're sick, because it's like 6 hours long. But we're curious. We're curious about how did we get here and where are we going? How can we help each other? How does this relationship even work? And does someone have to change for this relationship to work? These are things that we're curious about when we're watching this media depictions of human relationships. But in fact, all organisms engage in a diverse range of interactions with other organisms, ranging from the obvious predation and pollination, or even herbivory, even of highly toxic plants, right? To some that are a little less obvious. These are Acacia drepanalobium or Vicella drepanalobium, and they're ant mutualists. These plants make these domatia that the ants are housed in, and in fact, on one tree will be an entire ant colony that literally protects them from giraffes and elephants, right? This is a mutualism that we might not know as much about. Or this, which kind of looks gross to a room of not insect biologists, but these are butterfly caterpillars, butterfly larvae, and they are Fungaris is the genus name, and their entire life is dependent on host ants finding them in the wild, on the ground, finding them and taking them into their nest, where these guys parasitize the ant colony by being fed from the ants. So this is a highly specialized relationship that the lineage, the evolutionary lineage of this organism is dependent on a mutualism, a parasitic mutualism. And of course, everyone knows this one, I'm assuming in this room, with lichen. But then I also put this up, this is cyanobacteria, but really just to say that we're getting more and more knowledge about how bacteria is important in many ways in our world, from changing behavior and changing dietary outcomes, right? But if we wanted to ask those same questions in a biological framework, we might use a little bit more sciencey sounding questions. We might ask instead, what is the evolutionary trajectory? What type of symbioses are we? What mechanisms work to keep us together? And has there been any evolutionary innovation in this relationship? So I study that in Cycads. specifically because cycads have highly specialized relationships with both pollinators and herbivores. So these are one-to-one relationships where the pollinators live their entire life cycle on the plant, and the plant is dependent on reproduction on the pollinators. I'm going to talk a lot more about this. But they also have a lot of highly specialized herbivore relationships. These are Eumaeus atala butterflies that, as you can see, because they aggregate, they do great damage to the plant, and their entire life cycle is dependent on the host plant. But I also study cycads because they're just incredible. And I had someone reviewing a paper once, not at the review stage, but a collaborator reviewing a paper saying, you should really be careful and not assume that everyone loves cycads, which is great. And you should all think about that in writing your papers. Don't just assume everyone loves your system. But honestly, what is wrong with our world if you don't love cycads? So I'm going to take just a few minutes to convince you all that cycads are incredible plants. First off, they are just incredibly beautiful. These are photographs that my husband took at Montgomery Botanical Center. I put these in not just to show you the beautiful diversity, but also to point out that cycads are highly endangered, the most endangered organism, or the most endangered group of plants in the world. Most ranged from threatened to endangered. Just got louder. It's okay. Okay. And Montgomery Botanical Center is a conservation garden down in Florida that grows ex situ groupings of these plants. Here's just another picture that I love. But also, they also demand your attention in nature. So this is some field work I was doing in Panama, and just look at that. It's screaming for your attention, right? And the attention of herbivores, I should point out. But also, they have an incredible fossil record extending back over 265 million years, some with this incredible level of cellular diversity. So they are an ancient plant lineage, 265 million years. I always feel like I have to say that a few times to really drive this home. And there's lots of information that we can learn from this, from their fossil record that hopefully I'll learn more about with Liz, learning a little bit with her. But this is also just kind of interesting to me because the United States used to have a fossil national park, a Cycad fossil national monument. It's one of the very few national monuments in the history of the US that's ever been decommissioned, and that's because of theft of the fossils. That actually most of the theft was done by this George Weiland, which is this Yale paleontologist that really worked to establish the plants. So there's a lot of fossils of cycads and their related their related plants. So that's just an interesting side note that I love. But cycads are also thermogenic. So this means that they have their own body temperature. They have their own body temperature way of heating up. And they do this in their reproductive structures. This is actually really quite rare in plants. And when it is found in plants, it's usually that there is either one big burst of temperature increase or like an increase and then sort of maintenance at one level. Cycads, however, have an even more interesting pattern, in my opinion. They increase and heat up in their temperature over the course of the day, every day for about 2 weeks when the plant's reproductive. It happens in both the pollen cones and in the ovulate cones, and to a much less degree in the ovulate cones. This is something you might think of as like an animal trait, body temperature, but they also have another thing that people tend to think of as an animal trait. They have mobile sperm. They have flagellated sperm that are also gigantic. You can literally see these with the naked eye, which is a really fun thing to do that we should do in coursework, I think, here. But just to prove that they are the end-all be-all, they have an omega patterning in their leaf rachis. And I love this because I had this really crazy, and this is probably inappropriate to say, but slightly drunken night at Montgomery Botanical Center where I was talking to a girlfriend. There was somebody there that is an artist from New York, and she was there doing art based on sort of botanical representation in religious ceremonies. And she and I got all, a little tipsy together, and I was like, dude, what if cycads are God? And so they're not the alpha and the omega, but they certainly have omega patterning. But they are actually used, they have a lot of religious connotation in indigenous cultures. There's a lot of anthropology work that goes back and looks at things. For instance, the Aztec maize god, the Aztec corn god, they believe is sort of like a movement beyond an evolution from a cycad worship. They believe that cycad consumption could have been important for the Mexican Revolution. There's tons of great information about the history of cycads in that way. And they are still used in a lot of religious ceremonies, specifically where Catholicism has taken in some indigenous religious connotations. But something that's a little more important for our story is that cycads are super toxic. So they have two classes of toxins I'm just going to call BMAA and MAM. These toxins are mutagenic, neurotoxic, and carcinogenic, and they're present in all cycad genera and in all cycad tissues, and the effects of their toxicity have been recorded in insects and animals and even plants, which is amazing if you think about it, but that's a talk for another time. And this, of course, famously has been discussed in Oliver Sacks's book, The Island of the Colorblind, where he really connected consumption of cycad toxins with neurodegenerative diseases of the people of Guam. And in fact, this is still a very active area of research, is the toxin BMAA and its connections to ALS and Alzheimer's and Parkinson's disease. And so there's a lot of medical research that's still ongoing. They also have, besides their specialized relationships with insects, they have other specialized relationships. And this is some of the stuff that Chelsea first told me about that blew my mind. They have some specialized morphology to make their association with cyanobacteria. So they have these colloid roots, which you may not be able to see. This is a depotted plant, and here you've got sort of a colloid root growing up. Here's some, if you see in a root ball, me holding it. If you cut one of those root nodules open, you'll see a ring of cyanobacteria. This is some ongoing work down at Longhibio, and they're really working out that it's not just cyanobacteria that's present in these roots, that there are other bacteria, and that the interactions between those different bacteria can do different things for the plants. And of course, the hypothesis has always been, obviously, that cyanobacteria is fixing nitrogen for cycads, but actually that has not yet been proven. I, however, I think of myself as a symbiosis biologist, and I'm interested under this umbrella of symbiosis. I'm really a hippie, and so I really think a lot about mutualism, but nothing is just a pure mutualism. You may have a mutualistic relationship with your partner or your family, but trust me, there are some parasitic aspects of that relationship. And so in order to really understand mutualism, you need to also think about parasitism, right? And so in cycads, there are very clear distinctions that allow me to ask those kinds of questions. There's the pollinator mutualist that I'm going to talk a lot about today. And then I'm going to give you some insights that I've gotten from the parasitic herbivores that allow me to understand a little bit more of the relationship with the pollinators. And what I'm interested in here is mechanisms. I want to understand, how does this work? How does this relationship work? So I start by saying that specialized pollination is widespread in cycads. So for a long-standing time, we thought that cycads were wind-pollinated, even though the presence of insects had been found on the cones, but we now believe that all cycads appear to be obligately insect-pollinated. And in fact, it seems to mostly be Coleoptera, which is the order that includes beetles and weevils. It's found across the phylogeny, and some recent fossil evidence even dates that to before the rise of flowering plants. So we have a fossil that shows us where this beetle has already evolved morphology that is specific to its relationship with cycads and living within cycads. So I study the genus Zamia, which is neotropical. We've got one species native to Florida, and then the rest grow through the Caribbean, down through Central and South America, Mexico, and all the way down to Bolivia. And I study this because it's easy, because it's here in the US, it's much easier, especially because these are endangered plants and CITES regulated and all that. But also because of some great work done by two different groups of researchers in the 80s studying two different groups of plants. And they were working on the pollination of those plants and really challenging the idea that these are wind pollinated. Those researchers both found that they were pollinated by a genus of weevil, Rhopalotria weevil. And these were different species of Rhopalotria weevil. They found also that within a common garden, they were working at Fairchild Botanical Center and / Montgomery Botanical Center, or Fairchild Botanical Garden, that the weevil stayed faithful to their host plant. So one of those groups of researchers went even further and worked out the life cycle of the Rhopalotria weevil. And I'll just walk you through it, and then I'll show you some of my own pictures to give you a little bit more connection with it. So this is a large male plant with pollen cones all over it. The weevils aggregate on the outside of a pollen cone at a certain stage of development of the cone, a little bit before the cone opens. Then they begin feeding inside. They lay their eggs on it and on a microsporophyll, so I'm just going to call that a cone scale. So they lay their eggs inside the microsporophyll, inside the cone scale. The larvae develop. If they develop inside, hollowing it out. If one encounters the other, he cannibalizes it and then pupates in the stalk of the cone scale, where he then emerges and does the process all over again. Because of their pollination work, we knew that they visited the female plants. They visited the ovulate cones, but they never feed on them. They never aggregate there in large numbers. They don't lay their eggs on it. They don't develop on there. So this was still an unknown. Why? Why did they visit the female plants? And this relationship is so specialized and important, in fact, that the plants only have reproductive cones for about 1.5 to 2 months a year. So the rest of the 10 months of the year, the weevils are in diapause, which you can think of as like arrested development or hibernation, right? And think about this. This is in Florida. Nobody goes to Florida to hibernate, right? This is where people go to retire because it's comfortable. This is where, you know, Girls Gone Wild, spring break stuff happens. Nobody is going there to retire. But so for 10 months out of the year, these weevils find Florida too inhospitable for them because their life cycle is so tied to their host plant, even when there's a closely related host plant a meter away, right? So here's some of my own pictures of that. Here's a male plant. You can see there's a bunch of cones. It's hard to tell, but this cone here has a lot of weevils all over it. Look at them just aggregating like crazy. If you break this cone open, you'll see a lot of frass. That's weevil poop and nibble marks. Here's a weevil covered in pollen. And if you look at a cone and you open it up, you see a little weevil standing on top of a microspore fill with the pollen sacs above him. And then here's a female cone that is cracked open. So when a cone is receptive, it goes from being tightly bound to cracking open to receive pollen. So of course, in trying to understand this connection, why do they leave this house, this great home that gives them everything they need? All their friends are there, it's a party, there's food, it's like your mom's house when you're a teenager. And then you go out into the world on your own and you have to feed yourself, right? What did your mom do to kick you out of the house? How did we get them out of this house? What is this? What is this? So I think it has something to do with communication. And I have a little bit of reason to think that. There are two other really well-known obligate brood site pollination mechanisms, which means that the insect is housed and its brood develops within the reproductive tissue of the plant that it pollinates. So the ones that you guys have all probably heard of are the yucca and the yucca moth and the fig and the fig wasp. But I'm doing it in something that doesn't have a flower, which I think is particularly interesting. So I think that it has something to do with chemical communication. And that's my hypothesis, and that's what I started working with. And thank God I got lucky because it meant I got to get a PhD. So I started by asking, what is the plant signal? This is communication 101. This is what your therapist will talk to you about. What is your signal? What are you trying to say? How does your partner perceive the signal? And what is their behavioral response to the signal that you give them? And does the plant use that to enact reproduction? Or does your communication result in the desired behavior or the desired outcome that you're hoping for? So I start by asking, what's the plant signal? And for this, I do headspace collection methods. So here is a dehissing pollen cone. And I'm taking volatiles from it. So I have a inert plastic bag over it, literally a Reynolds oven bag, tied off. This filter here has absorbent material in it and a vacuum pulling over it. So for 45 minutes, I'm pulling the airspace around the cone over this absorbent material to get the smell that the plant is producing, the volatile bouquet, which I then elute and run on something called gas chromatography mass spectrometry, which is very simply just a way to separate all of the 10 to 15 compounds the plant may be producing so that I can ask questions about each individual compound. When I do that, I find that the plant that I'm studying, Zamia furfuracea, which is the one that had all of that great pollination work done already in it, the plants really produce two main volatile compounds. One that I identified as 1,3-octadiene and one that I identified as linalool. So that's the plant signal. The plant really only has two channels that it's communicating through, at least chemically. But that doesn't tell me what the insect perceives. So insects are different than humans. We can smell everything, right? Insects have sort of like a lock and key mechanism. They have proteins in their antennae that you could think of as the lock, and the volatile is the key that it has to come in and match, right? To close that lock, to fit into that protein in order for the insect to perceive it. So just the fact that the plant produces both of these doesn't tell me what the insect perceives. So for that, I do something called electroantennograph detection. So this is that GCMS that I talked about, what separates all of the volatile compounds, so I can ask questions about each of those. And what I'm doing here is I'm separating those volatile compounds. I'm sending half to be detected and produce that same image that you just saw, having the peaks at different times. But the other half is coming down this tube and being bathed across this prep. What this is two electrodes, and that's a weevil's little head that I just chopped off, I'm sorry. And his head is on one side and antennae on the other side. And so when the volatile comes and bathes across that, that's the key that comes into the lock, closes the lock, and it closes this electrical circuit, and it creates an electrical charge. And because I'm coupling that with the other analyses, I'm able to identify which of those compounds the insect perceives. So this is what that data looks like. You've got that exact same image over top, and on the bottom, here's my electrical charge, and then here's the dip. The weevil physiologically can perceive 1,3-octadiene and cannot perceive linalool, which is a much more common plant compound. But 1,3-octadiene is not very common. It's found in a couple other cycads, really, in Africa of all places. So this tells me that the physiological perception, and of course you have to do this many times, and to really identify this, you have to, this compound, there's many things that go behind it, but you really have to have a chemist synthesize this standard and test the weevil against that standard. And I did, as the same as linalool and all the different chimers of linalool. So we're positive that this weevil perceives 1,3-octadiene and nothing else that the plant produces. But that doesn't tell me what the behavioral response is. That tells me what the insect is capable of perceiving. So for that, I did something called pitfall tests. This is very basic. So I've got these Petri dishes with a hole cut in the middle and a pit in the bottom. And in the bottom, I'm putting different concentrations of 1,3-octadiene and a control, of course. And I put some weevils in the top, close the lid, put it in the dark so there's no visual signaling going on, and I come back after 30 minutes and see how many weevils have fallen into the pit, telling me, is this an attractant? A lot of compounds can be deterrents for weevils, right? And in fact, that you know this because you put different chemicals in to keep, you put chemicals in to keep mice out of the barn, and you put chemicals down to keep insects out of places, right? Chemicals can be an attractant or a deterrent. So when I do that, I see it, and do it a bunch of times, right? I see that it is an attractant. This is the number, this is the proportion of weevils that have gone down into the pit, and all of the different concentrations or amounts, base amounts of 1,3-octadiene. And what you see is that it is an attractant. Here's nothing in very low amounts. It is an attractant, but it is not linear. So you would think that just if you really liked something, you know, the more of it, the more attracted you are, right? Jesus, you said you love donuts, is that right? The more donuts, the more attracted you are. Do you get up to a point where too much donuts is unattractive, right? So it tells me, it tells me that this is an attractant, but not linearly, which is pretty interesting. So that made me think maybe this has something to do with what causes the movement from the pollen cone to the ovulate cone. And for that, I want to ask, can I elicit movement? Can I cause insect movement using these two different concentrations, which are ecologically relevant, right? These two different concentrations. So with that, I do something where I build these arenas. And so what you've got here is a box arena with a bunch of weevils in it. And the sides are all mesh so that there is airflow, so there's no like buildup of volatiles. On one side, I may put the volatile amount. On the other side, the 1,3-octadiene amount. On the other side, I put a control, I put nothing to see. And now I'm going to track these weevils over time and see if their location changes. Are they more attracted? Do they move towards the lower concentration and away from the higher concentration? This is what that looks like. I just always think it's such a fun video. There it goes, yeah. So I'm just tracking them over time. And of course, some various important things. If a weevil doesn't move at all, they're not counted. They may not be dead. They just, maybe this is the compound and they're happy. They may not be dead. But you can very easily do that using pixels, right? Understanding that like you take an average pixel density at this location throughout the course of the video and see how it changes, right? And when I do that, and I'm saying, I'm looking at their change in asymmetry, their change in direction towards the bait. I see that yes, at this lower amount, they are attracted. They move towards the bait. At the higher amount, they move away from the bait, right? So what does that mean though for how plants use that weevil behavioral preference for their volatiles to enact reproduction? And I had some knowledge, right? This is something that I had done in the lab. We know that cones will continue to undergo at least their thermogenic behavior, and even we know that cone scales are able to continue producing volatiles throughout the course of the day if they're broken off from the plant. So I had broken off cone scales from pollen cones and ovulate cones, 16 of each, arranged them in this arena, had it so that air could flow, all of this, introduced 4 weevils into like a circular arena with a pollen cone scale or an ovulate cone scale in the center. And then I just tracked them all day and said, how many are on the cone scale or within 1 little millimeter of it, right? And you can see from this that they're more attracted to the pollen cone scale than to the ovulate cone scale, and that there is this big dip in attraction in the pollen cone scale. There is also a statistically significant dip in the in the female cone scale. But of course, this is a lab setup, and so I don't know how much of this dip in either case is related to the fact that these are broken off cone scales, or what it may mean, right? But I do know that I at least see some difference. And I see a difference in attraction to pollen and ovulate cones, and also a big dip in attraction to pollen cones. So I go back to the plants and I take volatiles the same way I told you, but I do it every hour for pollen cones and for ovulate cones every hour, and watch how those volatiles change throughout the course of the day. The first thing I notice is that there is a characteristic pattern in both pollen cones and ovulate cones. So pollen cones are in blue here, ovulate cones are in red. What you're seeing is the percent composition of 1,3-octadiene. That's all I'm looking at now, not even linalool, which, you know, obviously, because it's the other compound, does an opposite shape. But what you see is that there's a real dip in production of 1,3-octadiene in the middle of the day, and then they approach 100%. So this is not overall amount that they produce. This is what they are producing. They produce these two compounds, and they shift to making only 1,3-octadiene towards the evening, a little after 8 p.m. And I can see also that pollen cones on average produce overall more volatile. So this is both 1,3-octadiene and linalool. How much do they produce? And I'm looking at total ion abundance, which is a value that I get from GCMS. Pollen cones again in blue. So pollen cones produce more than ovulate cones, and they have some peak at the end. Is that peak related to 1,3-octadiene? Very similar graph, but this is now looking just at 1,3-octadiene, not at overall volatiles. And yes, the pollen cones produce more 1,3-octadiene and have a big increase at the end of the day in 1,3-octadiene. Okay, so at the same time that I'm taking these volatile measurements every hour, I'm also videotaping cones. So I've got all these pollen cones, 10 of them, that I've broken off and put in these little plastic containers with airflow in them. And I videotape them all day long, and then just literally go and count how many weevils are in, how many weevils are in the container, which means out of the cone. And what I see is that at this time that I have this big increase, percent increase in production of 1,3-octadiene in the pollen cones, that's the same time that I have a big expulsion of pollinators from the pollen cones. So I'm counting, and it's like 2, 2, 2, 2, 40, right? So there's some massive movement. And I wish that I'm just not technologically advanced enough to be able to take like a 10-hour video and break it down into 30 seconds for you guys, because it's insanely striking. I mean, you're just watching and there's nothing, 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 and then it's just a million weevils, right? So our idea here is that I'm going to break this down and just use one pollen cone and one ovulate cone to make it a little bit clearer. But our idea here is that there is difference in concentration that is attractive to the pollinator, and that the plants use their daily patterning as a way to move the pollinators between the pollen cone and the ovulate cone. So it's almost as if there's this low level of production of 1,3-octadiene, and this looks like 0, but it's slightly above. That's just not really attractive to the pollinators. They just don't really care. It's not enough to get them to move or get them to do anything. But then there's a window that is attractive to them, an amount that they like, and an amount that they don't like, right? And the way I always describe this is, my husband, I love it if he has a little aftershave on, but I cannot stand cologne. So we've all been next to this person on the elevator, and you want to get off as fast as you can, right? That's the same thing that the weevils are doing, we believe. So just schematically, that would look a little something like this. There's a pollen cone here on the left, an ovulate cone here on the right. You have your increase in production of 1,3-octadiene sort of as a heat map. And you can imagine that over that daily cycle, there's an amount that is attractive to weevils. They're happy and they hang out in there. Then they get on the elevator with Mr. Stinky and they've got to get off. And the only thing that works for them, because their entire life is bound to this plant. This plant, that smell, is where they're going to lay their eggs. It's where they're going to feed. It's where they're going to reproduce. They're going to mate, right? Their larvae are going to develop. So they have no desire to find anything else. And so once they're pushed out of this cone, they're sort of pulled into this cone, sort of deceptively, right? A cheat mechanism, because there's no food here for them. And then maybe, and so in this case, this is still an unknown to me, is this one consistent amount? Or is there sort of a, it appears from this data, and it appears that what's happening is the female cone is making not enough to be attractive, but is becoming attractive at the same time that the pollen cone is becoming repulsive, right? But from the earlier data showing them in the lab, it seemed like maybe the female cone is being pretty consistent throughout the day. So that I think we should work out a little bit more. But this is actually called push-pull pollination. And it's been described in one other species of, or one of their genus, one species period, but it's in a different genus, Macrozamia. And Macrozamia have pollinators that are thrips, vastly, vastly different from beetles, right? So we know that this same pollination mechanism exists in Macrozamia and in Zamia. And we also know that that thermogenic patterning that I told you about produces a similar volatile patterning, like I'm showing. So we know that it's not just that an increase in temperature makes you more stinky. It's actually that the substrates that are necessary for volatile production are produced during the thermogenic process. And so you're able to test and see that it's predictable that there's a pattern where you have a peak in thermogenesis and about 10 to 30 minutes later you have a peak in volatile production. We also know that everywhere that it's been tested in cycads, every time it's been tested, we have this volatile patterning. And of course, we know that thermogenesis is ancestral in the group. So this really suggests to us that because every cycad has brood site mutualists, where the organisms live within the pollen cone, and that every pollen cone undergoes this thermogenesis that we know creates this volatile patterning, we think that this pollination mechanism may be ancestral in this group, and in fact may be a pollination mechanism that existed before the visual signaling that you get with the rise of flowering plants. So just a brief summary of that little section. We know that they only perceive 1,3-octadiene, and that it's both a repellent and an attractant, and that plants change that amount throughout the course of the day in a predictable pattern, and that can enact Weevil movement between the pollen cone and the ovulate cone. And this push-pull pollination mechanism is likely ancient and has existed long before the rise of flowering plants, potentially. But in still understanding mechanisms, I need to bring in this idea of parasitism, because these pollinators are herbivores of the plants, and the plants are super toxic, right? So in order to understand that, one of the first things I did was look at a phylogeny of Lepidoptera. So Lepidoptera is the order that includes butterflies and moths, and this may come as a massive surprise to you because it totally blew my mind as coming from a plant background. Butterflies are nested within moths. They're not two separate monophyletic groups, which is incredible. I expected gasps. So this black is butterflies, and all of the gray are moths. I've colored on the tips here where we have evolutions of Cycad-Weevil. In many cases, they are radiation, so they're genera that have radiated onto cycads and have, 8 to 20 species. And in most cases, so all of these guys, which include butterflies and a couple moths, these large herbivores that feed on leaves, there's aposematic coloring. It may not be as obvious in this one, but it is. We do not see any aposematic coloring here. Aposematic just means that they have warning coloration. They're like, I'm super bright red, don't eat me because I'm toxic. And we know a little bit about this one here, that it does sequester the plant toxins, that it uses them in their own body for defense against herbivores. These guys down here are all detritivores, which means that they feed on dead tissue. And so it's possible that they're feeding on dead cones where some of these chemical compounds, these toxins, may have already been leached from that system. Who knows what's going on with these little guys. But looking at this gives me the idea that Cycad-Weevil has evolved many times independently, right? And what's more likely? That they all got the same ability to feed on toxic cycads, or that they acquired some ability to feed on toxic cycads. And so for that, in order to understand the mechanisms, I thought I would look at the gut bacteria. We know a lot about what gut bacteria does and how it's related to diet. And so I thought maybe these guys all have shared gut bacteria. I guess these are my questions. So is there shared gut bacteria amongst herbivores of cycad tissues? And if so, can we infer anything from the function? So for this, what I first did was I took herbivores from, this is in 3 different species of Zamia and 3 different locations in Florida, and then 1 species of Cycas in Singapore. I took leaf herbivores, cone herbivores, and there's 2 different ones. This guy only feeds on the microsporophyll. This one only feeds in the cone peduncle. And then this guy that feeds on the stem, the underground stem. And I just very basically asked, what is the bacterial community of these herbivores? And is there anything shared? So for this, you do something 16S sequencing. You sequence all the bacteria in their guts and identify them sort of like ITS, like an I tag for bacteria. And then I just looked at it with a diversity analysis. And the first thing I see that stands out is that bacterial communities are generally distinct. So this is any individual of this species, its gut bacterial community is more closely similar to other gut bacterial communities of this same species. This is not surprising. This is totally expected. And we expect phylogeny to have an influence on gut bacteria. I can also look at the core. So that last image was every bacteria in the gut. So you yourselves may have 200 species of bacteria in your gut, right? But if I took the gut bacterial community of everyone in this room, and everyone had 200 species, and I asked, what's the core? What are bacteria that are found in the guts of all humans? It may be that there are 3 species, or 10 species, or 15 species that are found in every single one of us. And so that's what I did for each one of these. And the first thing you see is that 6 species here, and then large ones for some of these beetles. Also, again, not surprising. We know from the literature that butterflies in general have really transient bacteria. They don't have really a core bacteria. And beetles usually do. This is a little interesting. The other thing that's interesting to me is that these guys are feeding on leaves, and this guy is feeding on a microsporophyll, right? So it's a little more similar than some of the other tissue, right? Who's feeding on a stem and a cone peduncle. But when I look at all of those, I find that there are 5 shared bacteria amongst all of them, which is also super interesting when you think that these guys only have 6. So 5 of their 6 are found in all other cycad herbivores and not in related non-cycad feeding outgroups. So can I infer anything about this function? And yes, just a few things. These are the bacteria. We're able to identify 1 to species, 1 to genus, and then 2 to families, and 1 totally unidentified. And we can get some information about them. We know that these 2 both do nitrogen fixation. We know that Pantoea can be involved in toxin degradation and protein hydrolysis, which is particularly interesting because BMAA is a non-protein amino acid. And what it does is get into, well, one of the things it does is get into your proteins and change the conformation. But we also have this interesting anti-cancer capability. And in fact, this is used, this bacteria is found in earthworms and is used in Chinese medicine related to cancer. And it has, I'm gonna have to read it so I don't get it wrong, cytotoxicity and apoptotic and necrotic death of mammalian cancer cells due to the activity of a protein polysaccharide complex. So that gives us some interesting idea that maybe these bacteria are involved with the ability to feed on toxic cycads. It was interesting enough to us that we went and did this even further. So that previous study was published, but it was interesting enough that we did further. We have 12 species now of herbivores from all across the world, and this was your idea for this figure. It was great. It's beautiful, so I hope you all understand it. But so you can see there, we have all different feeding tissues and across different genera of cycads and from different geographies, and those 5 shared bacteria are still there. I'm working with some colleagues down at Longhibio. They're working out the function of these bacteria. So they have cultured insects' gut bacteria that I brought them from, they have cultured them in the presence and absence of BMAA, one of the toxins, grown them up over time. They have then separately, individually cultured bacteria and sequenced their genomes. And we're working just this week even on that publication. They're finding some pretty interesting stuff, understanding the function that these bacteria may be doing. But I don't have time to get into it today, so maybe for further time. So we know that there is a core microbiome of feeding on cycad plants, and then it may allow in some way, maybe it's just transient, maybe the cycads in the gut bacteria is selecting for this and has nothing to do with the insects, right? But this was the first evidence of convergence of a gut bacterial community across a diverse set of insects that share a common host plant. So if we go back to our questions that we're interested in relationships, we can ask them about cycads. And we can think about what is the evolutionary trajectory. And so this is something that I'm here working on with Chelsea. We know a little bit about the evolutionary history of cycads. And in fact, there's just this really amazing phylogeny where the lineages themselves are quite ancient, but the genera have all recently radiated. And we have a published phylogeny of Zamia, the genus that I study, just this last year. But we don't know anything about the evolutionary history of anybody else involved in this mutualism network. We don't have the evolutionary history of the pollinators, of the butterflies, of the bacteria. And so looking at an evolution of a mutualism network as a whole and not as individual players, and understanding how those relationships have evolved over time and how there may be correlations or what correlations may exist that we've been unaware of until now. And then understanding some of the morphology that may be associated with these relationships. What type of symbiosis are we? So it seems pretty obvious, right, that the pollinators are mutualists. They provide reproductive services to the plant. They're necessary. The plants don't set seed unless the pollinators are there. And they feed on essentially disposable tissue, just cone tissue that is going to die and rot away after the reproductive event anyways. But like I said, there's a little bit of parasitism in every relationship, which my husband doesn't like when I say, but oh my God, it's so true. And if you guys don't have kids yet, it gets even worse. But this is something that I'm working on a little bit further here with Rob Raguso in the neurobiology department. I just really want to understand from the plant's perspective, you need your volatiles to be static, essentially. You need to be able to, your pollinator has to be able to find you. And we've shown that that's really important. Volatile production is really important for that. However, you also need to hide from your herbivores. Because like I said, these herbivores decimate. They can decimate an adult plant, right? Nothing feeds on them because they're super toxic. And they aggregate in great numbers and eat a plant to the ground. So there's strong selective pressure in both areas, right? Sort of opposing selective pressures. But similarly, you have something going on with the insects. We know that these highly specialized relationships are sort of tenuous, right? These are, people call them evolutionary dead ends. So you don't, what do you do? What mechanisms exist that both maintain fidelity, the pollinator to the plant, but also allow for diversity, that also allow for you to change host plants or to find host plants, and that may explain diversification in both of these lineages, and maybe even explain some of this crazy phylogeny that we see. What mechanisms keep us together? That's what I really talked about today, and I think your therapist will tell you it's communication, and I, a symbiosis biologist, will tell you it's communication. You have to make sure that your signal is clear. Are you telling your partner what you mean to be telling them? You have to understand how they perceive the signal. Are they even capable of perceiving a signal and showing up and being there at that moment to understand what you're saying? What is their behavioral response, right, to the signal that you're producing? And does it give you the desired outcome, right? Does your communication give you the desired outcome? And of course, there may be some hidden diversity, and this is something we talk a lot about, and even something that Chelsea, with her new dean position, thinks a lot about. There's hidden diversity that allows us to show up and be present in this moment. And it is things that we don't know about and that in biology have been less tractable until now. But maybe there is something that is necessary before you can even show up and be a part of this relationship. And then, of course, has there been any evolutionary innovation? And I think that the really wonderful thing about systems like this, some systems that we would call a component community, is that, meaning that it's something where there's, Cycad-Weevil is a unique and sort of unusual food source and kind of extreme food source because of its toxicity. And in unique and extreme environments in nature, we expect a lot of endemics, and that's what we have. Cycad herbivores are almost exclusively, except for these weird detritivores, but almost exclusively specialized onto cycads and feed on nothing else. And when you have a system like that, it allows you to ask really wonderful questions looking at convergence. And you can see great examples in the literature where people have used convergence to understand molecular mechanisms and ecological mechanisms. And we're having some of the first evidence that there may be some sort of, I'm not even sure what you would call this, an ecological convergence of interactions. I'm not sure. But so because we have this great component community of not just the butterflies, but also the beetles and the weevils, we can ask questions about what it means to be a cycad herbivore. And with that, I'll just thank everyone. These are people that help specifically with this work. James was actually just here a couple weeks ago giving a talk. He specifically helped me with that behavioral arena and the tracking. He's responsible for that amazing video, not me. A lot of people that help specifically with the work. I really need to thank Montgomery Botanical Center. This is the conservation garden down in Florida. If any of you work on cycads or palms, please go there. They are highly dedicated to conserving these plants, but also to research. They are there for you and they do incredible work. I did all this work as a graduate student in Nomi Pierce's lab, and so you can see I was in a butterfly lab. But I studied plants, really. She studies species interactions. And then I just want to end by saying I'm on the board of the Cycad Society, which we send out quarterly newsletters related to cycad research, dedicated to conservation and research. And we even have some small grants for cycad-related research. And I brought some flyers for it. If anyone's interested in joining, it's a great society. There's flyers in the back and up here. So please come talk to me if you're interested. And that's it. I take some questions if anyone has any.
cyanobacteria, are they also on or in the cycad leaves, or are they just?
No, so that's some of the work we've done down at Longhibio. So they're specifically looking even at the coloraid roots, right? And we're not finding a correlation of them being on the plant tissue or even within the coloraid roots. Yeah.
So cyanobacteria produce BMAA, right?
Yes.
It's the BMA produced by the cyanobacteria move into a plant.
So that
modulates the herbivore's gut microbiome.
So there's still ongoing debate about if BMAA is produced by the plants or the cyanobacteria. So cyanobacteria is the only other place in nature that you find BMAA. And in fact, that's why it's so connected to Alzheimer's and ALS and that sort of stuff. I mean, they find BMAA in the brains of these patients. But there has been work where they've grown the plants in sterile conditions without the presence of cyanobacteria and find that they still produce BMAA. And even that there's sort of like a sequestering, I guess, to use an insect term, of BMAA in certain tissues, right? That would be more than the cyanobacteria could produce. So, and Dennis Stevenson, who's a good friend and on my committee, really strongly believes that there's been this horizontal gene transfer from that. And so, of course, I love him, so I sort of believe it. But I don't know. I mean, and you can probably attest the difficulty it would be to grow something without cyanobacteria getting into it, right? So that's kind of a problem with thinking about that work. But I don't know. So that the reason that we chose BMAA at Longhibio to grow the bacteria in the presence and absence of BMAA is because we thought that BMAA would have more effect on bacteria than the MAM glycosides, right? And we are finding interesting stuff, right? So the idea of growing bacteria in the presence and absence is saying, how do I watch this community of bacteria change over time? And with BMAA in the system. Are they using BMAA? Do we see BMAA change, its concentration change in the system? And we do. So understanding how exactly BMAA may have something to do with bacteria is interesting, but I don't have the answer for you yet. And of course, you know, there's potentially great medical implications for this, right? Because of the connection with Alzheimer's and whatnot. But I don't have the answer for exactly what it might be doing. Yeah, sorry.
I'm sure there's limited sampling, but in the species where headspace has been taking out the cones, does it look like 1,3-octadiene is a consistent signal?
So within my species, absolutely. It's in every one I've ever tested. And I've tested quite a bit across Zamia, and that's one of the things I'm doing here with Chelsea is looking at other phenotypes as well as the volatile phenotypes across the phylogeny. And it is not common in Zamia. It is found in it's found in 2 other species of Encephalartis, which are like super million years diverged in Africa. But in fact, one of the things that I didn't have a chance to talk about is I've gone further because I'm looking at this idea of coevolution. And so here's the one that I showed you. I'm just gonna call it the blue species, 1,3-octadiene. And I've done now another species because you have to use live weevils for this to cut their heads off and stuff. And so since there's only 2 that I can do that with, right? And so in this one, this plant produces vastly different chemical compounds, and the weevil responds to, this is methyl salicylate actually, and they cannot perceive the other host plant. So this weevil cannot perceive 1,3-octadiene, and this weevil cannot perceive methyl salicylate. So it is not common within Zamia, 1,3-octadiene, but it is common within the larger, or not even common, but it does exist within the larger group of cycads. So. Yeah.
So I'm not sure how to frame this question, so there's a lot of things going on in my mind. So it might come out incorrectly. It seemed curious to me that there is an order of magnitude difference between how much the ovulate cones produce versus the pollen cones produce. And what was curious was the weevils go out from the pollen cones, and at this point, is the low amount of octadiene produced by the ovulate cones, is it really an attractant, or are they just moving out because there isn't that much of the octadiene in the ovulate cones? Or is the ovulate cone producing something else that attracts the weevils to them?
Yeah, so I think that kind of gets at 2 questions. One is insect tracking and insect signaling, right? And so when you're an insect and you have to travel a long distance, your first connection is volatile, right? That's the thing that tracks you from a long distance until closer. And that's usually when visual signaling kicks in. And then of course you land and there's tactile signaling and gustatory receptors on the little legs and stuff once they get in. And remember, because these are dioecious plants, you should see them, they're like these big bushes, right? So the weevils have to get out of that. And I think they're sort of pushed out of that because of that smell. And then how they find the other one, they're likely still looking for the same smell, but maybe at a lower signal. But the cones, their only other tracking would be sort of a visual signaling to get to the female cones from that perspective. So that's one of the reasons I want to work with Rob Raguso, is to think about things. We know that there's thermogenesis involved, which means that there's humidity, and there's, even like thermal signaling, right, that's involved, that we would like to look at. It's possible that the ovulate cones are acting as an attractant at that lower level, right? Or it's possible that it's just completely by mistake, and that the pollen cones acted as an attractant at a lower level and a repellent at a higher level, and it's really all about men. But I'm like so sick of living in a world where it's all about men that I really want to believe that the woman's making a choice, right? But I don't know. I don't have that data to show, right? I have done like sticky traps near pollen cones or ovulate cones, and then sticky traps just sort of like in the world, right? And the ovulate cones do get more insects on the sticky traps than the ones that are out in the world, but it's also chaotic. You saw how many hundreds of insects are on a pollen cone. All of a sudden, it's just like a burst of weevils that are out and flying around. It could totally be by accident. But in nature, like that picture I showed in Panama, some of these, like, you're not showing up and seeing like a bunch of cycads really close to each other. They have to travel quite a ways, right? So how are they, right? I don't know. But it is true that in all cycads, that the females have a lower level of that in all cycads we know that the patterning is the same and it's at the same time, and that females produce less heat and less volatiles. Even if you're like correcting for size and stuff, So, yeah.
We'll take one more. I was gonna say, we're about out of time.
Oh, sorry.
We'll take one.
So I remember having read in some like pop science thing that it was like the thermogenesis that caused the weevils to leave the like pollen cone. Is that not a behavioral cue for them?
Well, so if you think about you're an insect, right? You're cold-blooded, right? So any heat is going to allow you to just warm up your flight muscles, which is going to make you more active. So it's the same as like a chemical process that you would do in organic chemistry. With heat, things just start like bumping around and moving around more. So I believe unquestionably thermogenesis has some cue for the insects. What's interesting is that you can elicit this behavior in both the thrips that were studied before and the weevils that I've studied here, regardless of temperature. And we also know that the temperature, if you recall, I showed that there's a temperature increase. It's about 30 minutes before the volatile increase. That we don't see the behavior really starting to happen until that 30 minutes, which of course like is a little window of time, honestly, right? It's not a massive window of time. But so I think that unquestionably thermogenesis has something to do with it, just because the weevils will become more active and they'll be more able to fly and more, because these guys don't fly really, and they even play dead when you're working with them. They just sort of like, I mean, really, it's kind of adorable, and they lay there for a little while before they get up. So temperature, there's no way that you can have such a strong signal as a plant and have it not have some impact. But the fact that you can create this behavior based just on that. And like I said, temperature also affects humidity, which affects all kinds of stuff, right? So that's a lot what I want to work out, is like, what are all of the signals? So, yeah, that's it.
That's about the time we have. So thanks to Shayla, and if anyone, she's around. She has her office up on the 5th floor, so you can come talk to her anytime. You're also welcome to come talk to her now. And thanks, guys.
This has been a production of Cornell University, on the web at cornell.edu.